All right, so we are going to be going into chapter 4 on sex determination and sex-linked characteristics. So, sex determination and sex-linked characteristics can be considered a very complex process. For As you see here, you see a picture of a dragon, specifically on the right side, what is called a bearded dragon. And what's interesting about these bearded dragons is that they are what we consider lizards that live in the desert or in Australia. And what was found interesting about these bearded dragons is that based off of the temperature in which they incubate their eggs when they lay them, will actually have an effect on their sex of each of the dragons that survive. Specifically, with bearded dragons, they don't exhibit the normal XY chromosomes that we would expect. They use Z and W. Not that denotes their shape, as we'll talk about further into the chapter, but ultimately they have the sex chromosomes where males are ZZ and females are ZW. And scientists discovered that over 20% of wild-caught females examined in 1 year had ZZ sex chromosomes, which are only expected to be in males. They found that when they incubated the eggs with the ZZ embryos at about 32 degrees Celsius, the offspring were all males. But as they increased the incubation temperature, they started to produce more ZZ embryos, which developed as females. So modulating the temperature, and I think that was about 36 degrees to produce ZZ individuals or females. So this is one way to understand, and I want to start off by talking about this because it helps you understand that sex determination is not just genetic, especially for some species, but it is also environmental, specifically dealing with the temperature regulation. And one of the issues or concerns that scientists have with looking at these bearded dragons is how their species may be affected by climate change. Because if the environment begins to change significantly and begins to warm significantly over time, then ultimately what you're going to have is the majority of the dragons that are produced, the bearded dragons or lizards, will be majority female. And eventually it will cause the species to go extinct because of that. So we're going to dive into this chapter. And this is one of my favorite areas just because understanding the complexity of sexual determination is extremely interesting. So there are several different mechanisms, as I stated, for sex determination. You have the X and Y, or the sex chromosomes, that pair during meiosis. And I'm going to preface my statement by saying they are not homologous chromosomes, but they share homologous regions or regions on the chromosomes that are necessary for sex determination, as you'll see when we talk about this. So sexual determination, or reproduction, I should say, alternates between what we call a haploid and diploid state, as I'll show you in the upcoming figure. And so ultimately what happens is you'll have a species going from haploid to diploid. But let's talk about what sex is, because I'm using the term sexual reproduction, sexual determination. What is that? Sex is used, the word or the term sex is used to refer to sexual phenotype. So most organisms are going to have two, male and female, okay? Male gametes tend to be smaller, female gametes tend to be larger, okay? And this is the mechanism by which we biologically determine or establish one's sex. Now, there are some differences when it comes to sex, and it has to do in part with some complications or abnormalities that occur during the embryogenesis and development process, where you can get different or abnormal sex types, which we'll talk about. But as you can see here in this this depiction here, you'll see that for meiosis is going to produce haploid gametes. Okay, one gamete is going to be haploid, and another gamete is going to be haploid, egg and sperm. But when they go through the fertilization process where they fuse together to form a zygote, then that zygote now becomes a diploid organism, because it has half genetic from one gamete and the half genetic components from another gamete, producing a diploid gamete. So what you're seeing in this diagram here is showing you the sperm gamete, or the sperm cell, that is, and we learned about this in the last chapter of the maturation of sperm, including the production of the tail. But you can see in this diagram that the sperm is trying to penetrate through the human egg, but the size between the two, the size difference between the two are significantly different. The egg is significantly larger than that of the male sperm. So one of the abnormalities, you can kind of call it, is when you're talking about hermaphrodism, okay? And this is where both sexes are present in the same organism, okay? So you'll have an organism that has both male and female reproductive structures, and they're said to be monoecious, meaning one house. If a species has one, and it produces either male or female, then they are considered to be dioecious. That means two houses. They do not house both reproductive organs for male and female. Humans are dioecious, and this is because males have male reproductive organs and females have female reproductive organs. So what role do chromosomes play in determining sex? This goes to the chromosomal theory of heredity, okay? And it states that genes are located on chromosomes. Now, why that is important is because they are the vehicles that are used for segregation of genes during meiosis. Remember, meiosis is the process for sex cells, okay? Stevens and Wilson showed that the X and the Y chromosomes separate into different cells in sperm formation. Half of the sperm receive the X chromosome and the other half receive the Y chromosome. Now we're talking about the X and Y. We're going to refer to mammals. But in other species, their sex determination or their sex chromosomes are different. For example, as you see here, in grasshoppers, their system is XXO, or the O meaning nothing, null. So this study was done by McClung with grasshoppers, and they called the system the XXXO system. And it's one of the simplest mechanisms of chromosomal sex determination. And this system, as you see here, okay, in the X06 system that you see here, females have 2 X chromosomes and males have 1 X. There is no O chromosome. That is just a place filler, as you can kind of consider it as a place filler, or the O will signify the absence of another X chromosome. Because the male, okay, produces, and then in females, you'll see that there is males have the XY and females have the XX. Okay, so XY for males, XX for females. Because males produce 2 different types of gametes in regards to their sex chromosome, not their autosomes, but their sex chromosomes, okay, they are considered to be heterogametic. Females, because they have 2 of the same XX, they are considered to be homogametic. So in looking at this Punnett square in this diagram here, it's showing the inheritance of sex in organisms with, specifically humans, with X and Y chromosomes. And you'll see that it results in equal number of males and female offspring. So you can see here's a male. He is exhibiting XY chromosomes, female contributing XX. Now remember, their gametes that they're carrying, the sperm for males or the egg for females, is only going to carry one. So it's going to be either, whether it's going to be haploid, it's going to have an X, or it's going to have a Y for males, it's going to have an X or another X for females. So if you were to set it up as a Punnett square, okay, this being the father on the top and on the side being the female, one egg is going to carry an X chromosome and another sperm could carry an X or a Y, but coming together they determine the sex of the potential progeny. Okay, and so if that be the case, it's about a 1-to-1 ratio of male to female production. So what role does chromosomes carry in sex determination? Remember, chromosomes are vehicles for carrying genes that help carry out and determine sex, that help regulate sex determination. So what you're seeing here is an X and a depiction of an X and a Y chromosome. Now notice the size of the chromosomes are significantly different. The Y chromosome is what we would consider acrocentric, and the X chromosome would be more synonymous with a metacentric. We talked about this structure of chromosomes in chapter 2. Now, what is interesting about them is they're not the same chromosome. The X chromosome is not homologous with the Y chromosome in terms of being the exact same chromosome, but they do have homologous regions on them. Okay, so even though it's not the same chromosomes, they share homologous regions. And typically on the red regions that you see here, which are on the tips or the ends of the chromosomes, are these homologous regions, and they are essential for sex determination. Okay, we call these regions the PAR region or the pseudo-autosomal region. Okay, the pseudo-autosomal region, and as we're gonna talk about a little bit more in a second, one of the essential genes in this pseudo-autosomal region or PAR region is the SRY gene. Okay, the SRY gene. Oh, let me go back. So in this region, you have the SRY gene that is important and significant for sex determination. And we'll come back to this in a few slides. But I will say, when it comes to crossing over, these two chromosomes do engage in crossing over. And if that region, the PAR region, is mutated, it can result in a sterile male. So these regions are very important, and crossing over is also essential for these two chromosomes, even though they're not considered homologous. So what you see here is another system. So remember, I mentioned to you a few slides ago that for sex determination, all species do not use the X and the Y chromosome system. Some are referred to as Z and W. Now, it does not mean that their chromosomes are in the shape of the ZW. That is just how they are determined. Okay, but we see here that in this case, in the ZZZW system, the female is heterogametic, which is different from the XY system. In this case here, the male is homogametic. That is because the male has two Z chromosomes, the female has a Z and a W. This typically goes with birds, snakes, butterflies, some amphibians, and fishes. Haploidy system. Haplodiploidy system is where you have a haploid set in males and a diploid set in females, and this is corresponds to bees, wasps, ants. Okay, so the genic sex determining system is not based off of the presence of sex chromosomes, but the presence of sex determining genes. Okay, the presence of sex determining genes. And in this case, scientists observed this in some plants, fungi, protozoans, and some fish. And what they found was that sex was not tied to the presence of sex chromosomes, but by individual genes. For an example, in mammals, You have the SRY gene that I just previously mentioned, which is located on the Y chromosome. Now, while that is a sex chromosome, it still has a significant weight in terms of sex determination. Okay, so in both genic sex determination and chromosome termination, sex is controlled by individual genes. So even in instances where species have sex chromosomes, it is still the presence of genes on those chromosomes that play a major role in determining sex. Outside of sex chromosomes and specific sex genes, you also have environmental factors that play a role in sex determination. Okay, a great example of that is using slipper limpets, which literally they stack on each other. So when they are larvae, they go and try to find an unoccupied area that they can begin to settle on. And so you'll first have one larva that will settle, and because it's the first one, it'll develop as a female. It'll develop or transfer to what we call a female limpet. And then it will start to produce chemicals that will attract other limpets or larvae to come in and settle on top of each other. And so what will happen is as they recruit, the limpet recruits other limpets to come in, the larvae to come in and settle there. They settle on top of each other, and the top is always going to be the male. So what you'll see is, as you see here, that first one becomes a female. Okay, it sends out those chemical signals to recruit other larvae to come. Other larvae come. The limpet actually then stacks on top of the female, and now what we have is a male. Okay, and it'll keep doing it. Eventually, the male on the top is going to switch sex, developing into a female to attract other larvae to come and settle on top to develop into male. So that's what you see here. Oops, excuse me. That's what you see here. Okay. But I want to point out that this is environmentally regulated as well. This would be considered, this type of sexual development would be considered sequential hermaphrodism. Okay, sequential hermaphrodism. And this is where you can have them kind of toggle between male and female. Not at the same time, but they can go back and forth between male and female. So I provided this table here for you to help with understanding some common sex determining systems. I would strongly suggest that you go through this and you pay attention to the system that is there, the specific mechanism for determining sex, male versus female, and the organisms associated with it. Is it environmental? Is it both? So going back and forth with this one. Now, briefly with looking at sex determination and fruit flies. The fruit fly has 8 chromosomes, 3 pairs of autosomes, and 1 pair of what we call sex chromosomes. Now, when it comes to what we call genic balance system, it's referring to having a balance between the ratio of the so-called what we call X and A. Okay, so the number of chromosomes to actual haploid sets of autosomes. And this was interesting because Bridges proposed that a ratio of an XA, a 1-to-1 ratio of X to A produces a female fly. And if that ratio is altered, it can produce a male fly. Okay. And there are some instances where you can have an intersex between a male and a female fly. So to really understand this more, Bridges began to examine other flies with different numbers of sex chromosomes and autosomes. And the XA ratio that I talked about appeared to correctly predict that phenotypic sex of the flies. However, as science progressed and they began to study this more, new evidence indicated that the genes on the X chromosomes are the primary sex determinant. The influence of the number of sets of autosomes on sex is an indirect effect, and it affects the timing of developmental events, and therefore how long sex determining genes on X chromosomes are active. So here again, you will see this table here provides the ratio, the XA ratio, in based off of different X or sex chromosomes. Whether you get XX, it's a 1, and you have 2 sets of autosomes, you're going to get that 1 ratio, and it's going to produce a female. However, if you have XY, 2 sets of haploid, and the ratio is 0.5, you get a male. If you have 1 X chromosome, 2 haploid sets, you get 0.5, it produces a male. But let's look down here for a metafemale, okay? That's where you have 3 X chromosomes, so poly X chromosomes, 2 sets of, autosomes, produces a 1.5 ratio, and it produces a metafemale. Okay, if you have more, in this case here, intersex is where you have more autosomes than you do the sex chromosomes, it will be less. The XO ratio is less than 1, and it produces intersex. So this brings me to that the SR gene that I was talking about. So how is sex determination affected, or how does it work in humans with the XY chromosome system? Okay. Humans, which are similar to the Drosophila having the XY chromosomes, the SRY gene is very, very important. Okay, this SRY gene is present on the Y chromosome, and it determines what we call maleness. Okay, it determines maleness. If there is anything that causes it to be imbalanced, it will alter sex determination. An example is the Turner syndrome. In Turner syndrome, you primarily see this in affecting females. It's where you have a underdeveloped secondary sex characteristic. So this is seen about 1 in 3,000 females. And typically when this happens, females are short, have a low hairline, a broad chest, folds on the back of their neck, and they tend to be sterile. Okay, this was founded by or discovered by Charles Ford in 1959. He used a new technique to study human chromosomes, and he identified that a 14-year-old girl with Turner syndrome only had 1 X chromosome, as you see here. Okay, and so it was referred as XO. The Klinefelter syndrome is another one. It 1 in 1,000, and it affects males. And this is where you have multiple males that have the Y chromosome, but multiple X chromosomes. Okay, males with this condition frequently have small testes and a reduced facial and pubic hair. They often tend to be taller than normal and are sterile. Most have what we consider to be normal intelligence as well. Polyfemale or poly-X females is about 1 in 1,000, so it's about similar to the occurrence is similar to the Klinefelter syndrome, and this is where you have females with more than 1 X chromosome. They call it triple X syndrome, where they have 3, but they can have more than 3. Okay, and in this case, they don't have distinctive features that other than the tendency to be tall and thin. However, they are sterile, and they'll still have the normal female process like menstrual system, and some of them can be fertile. So there's I don't want to say 50-50 chance, but there's a probability that some of them can still have normal reproductive options, but then there are some that actually can become sterile. However, the more X chromosomes that are added on, the severity of their intellectual disability increases. Okay, so this was the Turner syndrome. This is the example of the Klinefelter syndrome, where you have more than 1 X chromosome, but you still have the Y chromosome. So, given those examples, what is the role of the sex chromosomes? Okay, remember, these chromosomes are the vehicles for the genes that help to regulate and determine sex. The X chromosome contains the genetic information for both sexes. Okay, and so what we do know is at least one copy of an X chromosome is required for human development. There has not been a case where they've seen a normal human without any X chromosome. Okay, it's actually believed that if they don't have the any sex chromosomes, that they are actually aborted in utero. So they don't actually become a viable fetus. Another factor is the male determining gene on the Y chromosome. Okay, it's essential. And even if they have a single copy of this chromosome, even if it's in the presence of several X chromosomes, it still will produce a male phenotype. So that the male can have several X chromosomes, but as long as they have that Y chromosome with that male determining gene on it, the SRY gene, they can still produce a phenotypic male. Okay. If a embryo or a zygote has X chromosomes but no Y chromosome, it's going to produce a female phenotype. Okay. Genes can affect fertility are located on the X and the Y chromosome. A female usually needs at least 2 copies of the X chromosome to be fertile. So even if a female has one X chromosome, they can still become a female, but they need both X chromosomes to be fertile. So this brings us back to the SRY gene that I was talking about. So you'll notice here on the Y chromosomes, we have those regions that we talked about previously, okay, that are essential for sex determination. Those are called the PAR region, the pseudo-autosomal region, in red here, as you can see, okay, the pseudo-autosomal regions. Now, on this acrocentric chromosome, the Y chromosome, you have the presence of the SRY gene, which is important for male development and to produce a phenotypic male. If any of these regions this region is abnormal, it is not able to undergo crossing over, it will result in a sterile male. Okay. A sterile male, and in part is because this gene helps to promote the differentiation of the testes. Okay, so it's so important, and also plays a role in the fertility abilities of males as well. We also have the androgen insensitivity syndrome, and this is caused by the defective androgen receptor. Okay. So outside of the SRY gene, there are other genes that are essential for sexual development. Okay. And we see this is developed in women with the androgen insensitivity syndrome. These individuals have female external sexual characteristics, and most of them are not even aware of their condition until they hit puberty. Okay. When examined by a gynecologist, they will actually see a vagina, but it's going to end blindly. And then the uterus, the oviducts, the ovaries are all absent. And inside of the abdominal cavity of women with this issue are a pair of testes that produce testosterone, like you see in males. Okay. When looking at the cells, the chromosomes of these individuals with this disorder, they have an X and a Y. Okay. Now, how is this possible? How can a person appear as a female when her cell contains Y chromosomes, right? You have to look at the relationship between the genes and sex in humans. Okay, so in a human embryo with the Y chromosome, the SRY gene causes the gonads, okay, to develop into testes, which produce testosterone. Normally, you're going to see testosterone stimulate embryonic tissue to develop in male characteristics, but what you see in individuals with androgen insensitivity syndrome is they lack the receptor. So even though the testosterone is being secreted, it's not being able to activate the pathway because the receptor is absent. Okay, so this also tells us that this androgen receptor is necessary for male development. Otherwise, it's going to result in a phenotypic female. Okay, and this receptor, the gene for this receptor, is located on the X chromosome. So people that get this condition are going to inherit it from their mother. Okay, people that get this condition are going to inherit it from their mother. All right, so with sex determination being linked to other genes, or sex characteristics are linked to other genes on the sex chromosome. If the genes are located on the X chromosome, it's considered X-linked characteristics. If it's located on the Y chromosome, it's considered XY-linked characteristics. This is something that we can attribute to Thomas Hunt Morgan. Okay, he was the first person to really explain sex-linked inheritance, and he did this using Drosophila, or fruit flies. He actually built his career off of studying these experiments on fruit flies. Okay, he discovered that among the flies in his laboratory, there was a colony of males that possessed white eyes amongst this cohort, or this large number of female flies that were containing red eyes. So he began to investigate this further because he wanted to understand, why did he have these small amount of males that had white eyes compared to the large amount of female flies with red eyes. So his study, he began to cross pure breeding red-eyed females, okay, as you see here. And in the let me just point out, for fruit flies, if it is a wild type, or wild type being normal, you're going to see it categorized or denoted by a plus sign. That means normal, or what we consider wild type. If it is mutant, then you'll see in this case here, it's still going to refer to the chromosome where it's located, but then it'll have a initial there, in this case for white eyes. That lets you know that it is abnormal. Okay, this is the first chromosome. This is the second chromosome. Okay. So in this case, he mated, okay, or he crossed pure breeding red-eyed females, which is what you see here, 2 X's with a plus sign, with a white-eyed male, producing an F1 progeny that all had red eyes. Okay. Male and female all had red eyes. Okay. Red eyes, red eyes. Okay. So his results from his initial study were similar to that of Mendel. And so he really did build off of Mendel's study, and it really did it confirmed some of what Mendel saw in his work for the initial F1. Okay. So he then he continued this cross by taking the F1 progeny, okay, and crossing them. And this is where the white-eyed males came up, or showed up in the F2 generation, by crossing them. And this is what Mendel also saw. Okay. But now what was interesting is that his results suggested that white eyes are a simple recessive trait, right? But when he began to do a second round, it showed up, but only in males at a smaller rate. Okay. So Morgan proposed that the locus affecting eye color is on the X chromosome, because it disproportionately affected males to females in terms of white eye color. Okay. So because of this, because it's on the X chromosome, eye color, males cannot be homozygous or heterozygous, but they are said to be hemizygous. Hemi meaning half, because they don't have 2 X chromosomes. So they don't have the ability to be homozygous, meaning having 2, and they don't have the ability to be hetero because they technically don't have, again, 2 X chromosomes. So the proper terminology for males when it comes to X-linked traits or Y-linked traits would be hemizygous. And let me point out too, X-linked color and blindness is also seen in females in humans as well. So to further understand this phenomenon of this small amount of males showing up with white eyes, and in comparison to the stark amount of red eyes in females, one of Morgan's students named Calvin Bridgers, he set out to investigate the genetic basis for these exceptions, okay? Because ultimately, he kept experiencing this unexpected phenotype to appearing in these crosses that he was conducting. And then initially, Morgan thought it was a random mutation, and Bridges did not believe so. So he began to do some studies with the flies, and what he concluded from his studies when he mated white-eyed females with red-eyed males, and this is what we would call a type of reverse type cross, okay? He found that the white-eyed females and red-eyed males in the F1 result from non-disjunction of the X chromosome in a multi-X chromosome female. So he was able to determine this abnormality because what we call non-disjunction. So a portion of the Y chromosome was actually attaching to the X chromosome, and it was allowing for it to almost resemble that of males, okay? So this is where Calvin Bridges' explanation or his results came from, and he actually confirmed what he predicted. And how he did that was he examined the chromosomes of the flies that he found from his study, and he found that what he actually predicted. So this was not a random mutation, okay? It just didn't randomly happen, but it was actually due to non-disjunction. All right. So this is just looking at what we call Z-linked characteristics, and this is a cameo phenotype in Indian blue P file as an inherited as an Z-linked recessive trait. So you'll see that blue females, okay, the blue females were crossed with a cameo female, which you cameo male, as you see here, and then in the reciprocal cross, it's just the opposite. You had a cameo female crossed with a blue male, okay, and they looked at the result of doing the opposite or cross-linking the two, okay. And what they found was in organisms with the ZZZW sex determination, the female always inherits her W chromosome from her mother, okay, and she inherits her Z chromosome along with any Z-linked alleles from her father. In this system, okay, the male inherits the Z chromosome along with any Z-linked from both mother and father. So this is kind of the opposite of what we see in the XY sex determination. All right, so this is going to be all for that I'm going to cover in this lecture, but make sure you go through and read the entirety of chapter 4.